There we go. Hello.
Hello, how are you?
Pretty good.
Great. Looks like Sushmanth and Himanshu are here. Welcome. How are you doing, Susan?
All right. I haven't got my graphs done that I need for 2 o'clock today, so just
yeah.
I need to hurry up and do some of them, at least some of them.
Yeah.
I could do a dozen graphs with my data, so.
Oh wow, great. But the minute you have to give a presentation?
Yeah, I have to say, see, I did this. So it's better to have graphs than just data, because the data you can go, I think it's increasing. Yeah. Anyway, it's forming J curves, so that's nice.
Yeah.
Well, I don't know if they're exactly J, but anyway, they're curved nicely.
Great. Sounds great.
And I have those eggs.
Oh, okay. Yeah. Great. Yeah, I heard about that. So, yeah.
Salamanders laid eggs.
Yeah.
News of the century.
So congratulations to Sushmanth and Himanshu. They're both GSoC students officially. So it's great.
Wow.
Yeah.
Congratulations.
Thank you. Hi, Bradley.
Hi, how are you?
I hope you are good. Fine. Thanks for selecting me.
Yeah.
I couldn't mention how happy I am. Even my father was more happier than me.
Yeah.
Because he wanted to get selected. Yeah. I couldn't mention. Last 2 days was full of he was enjoying all the stuff he's working on. I have worked a little bit last 3 days. Actually, I was having a company interview, not this internship, company interview for a full-time offer. I want to give my updates. Is it possible? Can I share my screen and explain about my work for next few weeks? I made a presentation actually.
Okay, yeah, it's not great. Please share your screen.
Where do I start now? Right now, I was actually working on two things. Instance segmentation. We have this mind. I have been discussing this with Mayuk Dev. He mentioned that actually he want to attend today's meeting due to some other conflicts. He couldn't attend the meeting. We decided to train the models on cell tracking challenge dataset. Wait a minute, I will provide a link of it in the chat box. This is the dataset I am using. There are 3D dataset from here. This has the ground truth of the membrane segmentation and the location of the nucleus also. So as per mentioned in the proposal, like I don't need to create some segmentation editor to make those all ground truths. Actually, we can use this dataset and train the model. Right now, I want to divide my work into two phases. Sorry, there is my slides.
Yes.
Yeah. Okay, I want to divide my work into two phases. One was the model which is trained on SAM model, segment anything model from Meta. I am going to fine tune this model and train the dataset on the C elegans and then use it for instance segmentation. And this will be my first phase of work. And the DevoNet which I have mentioned in the proposal will be my second phase of work. This is going to take just 2 weeks to train the model and fine tune it, and we can make two models actually. One was cell membrane segmentation, only the membrane. Actually, DevoLearn, I didn't understand this. I want to discuss with my. Sorry, I'll make complete PPT and I will share it to you in next coming meetings. And this is DevoLearn. Cell membrane segmenter. Here in cell membrane segmenter, we are extracting the centroids. I don't understand what these centroids are exactly, because cell membrane segmenter file should be only segmenting the membrane of the cell, and so here they are extracting centroids also. We need to extract the centroids in nucleus membrane segmenter, in nucleus segmenter actually, cell nucleus segmenter. Here we need to extract the centroids and the volume of the cell. But here they are doing in reverse method. I didn't understand that. So I was thinking in my plan of action while using SAM. Sorry, where is while using SAM, I'm gonna make a cell membrane segmenter which only segments the membrane, and it's gonna useful for you in the future also. You mentioned that you are keeping something in mind and making this all membrane level segmentation. Due to that, I'm gonna make this cell membrane segmenter only for segmenting the membrane part, and the cell nucleus segmenter separately. With the SAM model, I'm gonna fine tune it and extract the centroids and the volume of the cells, and this will be the SAM, and another one will be the DevoNet, which I'm going to use nucleus segmentation network and nucleus detection network. I'm going through the data set actually. This is code. I wrote a little bit of code. I'm making the notebooks, and I'm going to keep the sorry membrane. The repo in this format. Actually, I'm going through segment anything model. They are like well structured format. Our DevoLearn is also good, but people when came to DevoLearn, they can't understand what it really is. So I'm providing some notebooks which can explain a little bit about what we are doing, and how to handle the data set of cell tracking challenge data set. Everything will be cleanly explained, starting pre-processing, building a model, fine-tuning it. Every different notebook explains the different process of these all things. And yeah, that's in one repo only. I will keep a segment anything model trained on C. elegans and another DevoNet also in the same repo. If someone comes reads the repo, they easily can understand what is going on in the repo. This would be with the file structure. And actually, I mentioned that Fiji there is a platform. This is the platform actually Image. Last few days I was thinking to use this to create the ground truth for every cell. But luckily, when I was going through the data set of cell tracking challenge data set, I could see there are ground truths I already mentioned in the cell tracking challenge data set, and the nucleus position also was extracted there. All ground truths were before only created. Mike told us to work on this thing, like he give a green signal to work and start implementing. Right now, I'm creating a pre-processing file, adding different colors, what is the file structure, and adding explanation to it. Next 2 weeks, I will be working on the model. Parallelly, I was writing blog to easily understand, and those will be uploaded in the DevoLearn blog section. And that's what is there in my mind actually.
Well, that's great.
Yeah. I will share the slides when they are completely full. Due to this interview process,
right.
I was kind of busy and I couldn't done. After, I will make slides and keep it in the DevoWorm Slack channel. So if anyone didn't attend the meeting, also they could easily understand.
Great. Yeah.
By 9:10. Sorry for the incomplete work.
Oh, no problem. Yeah. Yeah. So I mean, this will be the thing that you'll do like every week in the summer. Like you'll just give a quick update, and you don't have to have a complete talk, but just show what you've done. So that's good. Good start. So yeah, I like the idea of using Fiji as like a kind of a benchmark.
So actually, there is segmentation editor in this. With that, we can create the ground truth for every cell manually. And so if we create ground truth for at least 10 to 15 cells, that would be enough for training, and another 5 other video cells we can just generally use it for validation. Testing it would be easily. But for SAM from Meta, that is not useful. Training at all not useful, because it was already trained on 11 billion images of cells of different things. So, and it's really not needed to train it, but I am thinking to train it, fine tune it for particularly to DevoLearn, and take out the ONNX from that and upload in Hugging Face as a app. We have 3 models in Spaces, right? Like that only I will upload this instance segmentation in Hugging Face also. There is actually support for ONNX, support for this SAM model, segment editing model.
Yeah. That's great.
That's okay. You have any doubts or questions? Could you, is there any?
Yeah. So I mean, no, it looks good. I mean, it's definitely putting in different tools that exist, because everything's moving pretty fast, and we always like to improve upon what we're doing. And I like the idea of using Fiji as sort of like a benchmarking tool. Fiji, of course, is sort of a version of ImageJ that they've open sourced. I think they've specialized it for various functions and things like that. But it's yeah, so it's got a good connection to the what people are doing in cell biology. If you're working in cell biology and you're not really interested in the computational part, you get ImageJ and you segment cells, and you can do like a decent job if you have some knowledge of what the what you're trying to, the questions you're trying to ask, I guess. We're kind of a little bit farther away from that. Like we want to be able to, discover things. So yeah, and then of course there are all sorts of algorithms you can use that are more extensive versus what like Fiji and ImageJ have to offer. So yeah, this is your notebook.
This is my code actually, pre-processing code.
Okay.
These are things I plotted actually. This is the segmentation masks, and I created this 3D tool also to visualize how JPG compression loss is there.
Okay.
This is for JPEG 3D visualization of when we convert a TIFF file to JPG. I'm just making all these visualization tools to easily understand. When someone visits it, I'm adding some documentation to it also. That I'm making in a phases way, like start we need to pre-process the data, a complete explanation of it, next segmentation model, next fine tuning, etc. The process will go on like this. This is the part of code I worked on this week.
Yeah. So a bit about the loss functions there. So why does it have that kind of profile, do you think? I mean, why is it not evenly distributed across the image?
Yeah, I mean that is due to fluorescence effect, I think so. Okay. Some parts are, when you see the upward view of it, you can see this part is like has good imaging, but these cells doesn't have good imaging. This is a 3D file, right? When you see from upward, this is actually a C. elegans photo, which was seen an upper view, and this is the 3D data set. I mean, you can see, right, this kind of tables.
Yeah.
I mean, each represents intensity or the birth time, because maybe this cell would be from starting onwards. Due to that is the reason this has the highest height.
Right.
I mean, it started dividing from there itself. It has the highest height. This is how we can calculate the volume of cell also, because we are having the height area of the cell from upward. So we can calculate the volume of each cell, number of frames into total number of frames into area of the cell. So that would give out the volume of the cell.
Yeah.
What is a typical volume for the cells?
I couldn't. Can you just repeat the question? There is no particular like ground truth for these volumes of each cell, but this was Mayuk's idea actually. He told to take number of frames into the area of cell. That could give the volume of cell, because it's a 3D data set, right? That way we can extract the volume of each cell he told. So I was trying to implement his idea only and extract that.
Right. Yeah, that's good. Yeah, I don't think that I think at that stage of development, I think it's C. elegans, there isn't really I don't know, I guess there is a characteristic volume, but I don't think there's a lot of variation there. So, I mean,
I'm actually trying to find some sort of a volume for my model.
Oh, okay, for the Axolotl.
Yeah.
Or for the tensegrity.
Yeah.
Oh. Yeah.
I also have these images of eggs, so maybe, maybe Axolotl could be included in some of this. I don't know. I was a bit disappointed with my images. There's a couple of ways I can improve them, but they're slightly blurred. I'll show you later.
Yeah.
And another thing, I'm creating a repo in my own account for GSoC 2023 work.
Okay.
After doing a couple of works, then I will push the whole code to DevoLearn. Or
Okay.
you will create a repo in DevoLearn, or should I contribute to it? How could I do?
So this is for like, what is it? For something specific, or like, is it just for the GSoC work? I was planning on.
GSoC.
Yeah, I'll create some repos for GSoC work,
Okay.
and then we'll kind of go over those weekly. Like, I just want to have a place where we can do GSoC work, and then we'll either push it or clone it to somewhere else. So a lot of the work will probably end up in the DevoLearn organization, and so I'll probably set up a repo in the DevoWorm organization, or I guess the DevoWorm account. It's not really an organization, but just to have it so we can review it on a weekly basis.
That's what my updates are. I will update with the blog section. So that's what I was thinking.
Okay.
This week. So yeah, that's it.
All right. Well, thank you. Yeah, that was a great update. So we'll set up the repositories closer to the coding period. And I'll go over in a minute, like a lot of the details of what what we'll be doing here. Himanshu, did you have anything to update us with yet, or?
No, sorry to interrupt. I have one thing. I was thinking to extend my project period. I want to work in a longer period to make this project successful as possible. I was thinking to extend it by 20 weeks. I mean, by how much end it is there, I want to do it on that time. Because I have the alternate version for imaging. Fiji. Yeah, this is the alternate version. Sorry.
Yeah, it's this Fiji. I'll put it in the chat. Or Sushmanth did. So yeah, I mean, you can extend. I mean, we'll talk about that in a minute. There will be like an official period where you have to submit something.
Actually, we got a mail. Actually, you need to update in some web app to extend the time period. They have mentioned in the GSoC mail. Okay. So that is the reason I'm mentioning in the handoff. I want to work a lot of time on this project clearly to create the complete workflow, everything. So I want to take how much time I have to do this project.
Yeah. Okay. Yeah, we'll work out the details of that later, I guess. But I think I'll talk about that in a minute. Okay. Yeah, Himanshu, I don't know if you wanted to say anything.
Hello, am I audible?
Yeah.
Oh yeah. Hi, Pratik.
Hi.
And hi. Okay, I'll just share my screen.
Okay, go ahead.
Yeah, is it visible?
Yes.
Okay, so yeah, basically I have a really short update. Like basically I had a meet with Mayuk like yesterday, and he gave some advice about some starting points, and he talked about my approach and the way to do things. So like one of the things he mentioned was, since it's the starting stage of the project, you have to keep a final goal in mind. So some kind of a long-term plan that could be implemented. So one of the main things that was given in the proposal as well was keeping DevoGraph as a sub-module in DevoLearn. So the DevoLearn, because it's DevoGraph takes a lot of things from DevoLearn as well, and we have to just create a tool which incorporates both of them at the same time. Another few things that were mentioned, even Sushmanth is doing on this, restoring DevoLearn model weights to Hugging Faces, and one of the other things that were mentioned was to keep DevoLearn as well on the side, but whatever work we'll do this summer, we can store the model weights, or we can just import the model to Hugging Face itself.
Right.
Another thing was since it's a tool that can be used by everyone, and ease of use is something which we can keep in mind. another thing that was like it's a good idea to implement is to create a web app or interface or a basic neat CLI tool, like using something like Pilot, for example, to get some ease of use instead of like the way we use in DevoLearn. Like, so someone who just wants to do it, like just wants to do segmentation and doesn't want to work inside the code of things like someone which can just use like the Fiji tool or some TD analysis tool, like some kind of tool which is like easy to use. So some things to keep in mind. And another thing which was like to change was to change the way the library was written itself, like DevoLearn, like to refactor the existing CNN models as well as getting some new models inside. So like for this, like a particular code of a particular way of doing or implementing the code should be done, like it in the refactoring stage. So also like to move the models to Hugging Faces for future proofing, like anything that you have created, like for example the current segmentation models as well as the model like Sushmanth is doing, and etc. So basically there are two ways you can like work around with Hugging Faces. Like you can just upload the PTH file, or you can just create a model inside Hugging Faces, like using their way of doing things, and both of them is like viable options. Also, another thing is like a kind of like a final idea would be to revamp the repo, or revamp the existing DevoLearn or DevoGraph repo, or else create a new repo in which there will be like some kind of tools or utilities, like for example, if someone wants to do just a particular things on like a particular part of a data set, or particular he just wants to segmentation, or he wants to do stuff with like what you're doing with graph embeddings and GNNs. So basically a CLI tool on top of the models which you are creating. So the advantage for doing this is that if someone wants to get inside of things or change the model we are implementing, they can do that. But if someone just wants to use the utilities, they can do that as well. This was kind of mentioned in this particular issue, like DevoLearn issue number 55, or like in the long-term vision, where there is a input pipeline which relies on the base inference engine, which has the model and pre-processing and the transforms. It relies on this, and the non-programmer will just use a web app or a UPI, web app or a GUI, and they can just use the models which you are using. After that, like one of the things like I was going across was about C elegans itself, because it's something which is new to me and I wanted to learn more about it. So I watched some YouTube videos, and one of the things which which I came across was someone like Sidney Brennan itself. Like Sidney Brennan, they discovered messenger RNA, and they also helped solve genetic code. And after that, they moved on to development of the nervous system, in which they wrote a paper about the ideal model organism, that is C. elegans. And this is something which was interesting to me, like why C. elegans was used, and why it is like a good model organism for developmental biology. So this is some advantages of C. elegans, which is just notes. And I also went across the onboarding guide, which was given to you, which was given to us on the mail. Yeah, which has a lot of information. I'm still going across this, but like this particular structure, and like it's a lot of things, and this repo as well is really good.
Yeah, we have. We've used that in years past. This part, the model organism biology, is useful, I think, but we'd always like, I think we talked about this last week. We always like to keep it up with the latest stuff. So if you find something that you find particularly useful, you could add it in there if you wouldn't mind. as a pull
Yes,
request. Yeah.
yes, I would love to. I'm still in the research phase and figuring out how to keep up with this, and but I'll catch up soon.
Yeah.
Yeah, but this is my update.
All right. Well, looks good. Yeah, and then you did have a presentation on your proposal, but as I'll say later, you know, we'll try to do that like in a few weeks. We're gonna have like a
Yeah.
presentation on proposals. So
I'm working on that. Like since I had a meet with Mayuk as well, like I had to take into account their ideas and add more ideas to put into. So it's still work in progress.
yeah,
But the current gist of the things I've proposed in my proposal are still there. Like what could be done.
right. Yeah, so that's great. That's great. Thanks for the showing your stuff there. Yeah, and then it looks like Jiahang is with us. Thank you for coming, Jiahang. And Jesse's here, and Dick is here. So let me start. I know Sushmanth had to leave. He was having connectivity problems. Okay. So I want to go over some materials for the community period because this is the beginning of the community period. So we have the two people we've selected, Sushmanth and Himanshu, and this period from now until the end of May is the community period. So what does that mean, and what are we going to do with respect to our group? So the community period is generally for people to get familiar with the community. And by community, I mean like the group you're working with and some of the other resources that exist. So we are the DevoWorm group. We've been around since about 2014. We've had collaborators come and go, and so we have done in the course of that time a large number of collaborations, GSoC projects, and the like. So we have a lot of things that have been around over time. Now, we also are affiliated with the OpenWorm Foundation. So the OpenWorm Foundation is interested in the C. elegans model organism. The idea there is to create a digital C. elegans with all the different parts. So we're the developmental aspect of that. And they have, the movement group. They have the connectome group, the physiological modeling group. There are people who do biophysics and so forth. So there are all sorts of different groups within OpenWorm that are doing things, publishing papers and the like. So there are a couple of resources I wanted to go over. So the first one is the timeline. So we are, this is the timeline for 2023 Google Summer of Code. We are right about here. So we're at the beginning. People were selected on the 4th, which was Thursday. And then if we go out to May 28th, from now until May 28th, that's the community bonding period. So this is where you can work on, getting your projects in a row, but also you'll want to do things like become familiar with C. elegans, or you'll want to become familiar with the tools you're going to use. But you also, on top of that, want to get familiar with your community. So I know Sushmanth has been working in the community here for a while, so he knows a little bit about it. And then, of course, Himanshu has been working a little bit. He's kind of familiar with it, but I would encourage you to reach out. I know you've been exploring the onboarding guide, so these are things that you can do during the community period to get a sense of what the opportunities are in your project. So that's the community period. I'll also maybe provide resources on open source software creation and how to do that. And so there are a number of things we'll do over the community period. Like I said, the other thing I want people to do is to take your proposals and turn them into a short presentation. And so, whatever you did for your proposal, you want to create a short presentation, maybe 15 to 20 minutes, maybe a little bit less, where you go over your proposal and you kind of stake out what you want to do. And then I want you to present that towards the end of the community period. So it would be in a meeting sort of at the end of May, early June. I'm not really sure what the timing of the meetings are right then, but once you do that, you'll have that presentation, but you'll also then revisit that presentation at the end of the summer when you are finished with your project. And the idea there is to take the slides that you, like maybe the things you didn't do, or the things that you changed, and then add new slides based on what you've done. So it'll be like a new version of that talk. The reason I do that is because I want you to understand, I want people to understand how these things change over time from the proposal to the finished product. So I want people to do that. And so if you have any questions about that, let me know. But I think the first part is pretty straightforward. So then we go to August. I think it's September. Let's see. I think actually August 28th is where the last week of projects ends. So the final week, GSoC contributors submit their final work August 28th at 1800 UTC. So that's the end of the project. Now you can request an extension on your project, which is good because, sometimes you have things that come up during the summer. I've had people who have had exams during the summer and they need to take a week off to study, and that's, we can work around that. But, generally, we shouldn't abuse the extension period. So I don't want to, kind of use that as sort of a way to procrastinate. There is one evaluation in the middle of the period. That is July 14th. So around July 14th, I'll be evaluating your work. And it's not a big deal. As long as you're doing your work and reporting and coming to the meetings and we know where you are, that's all that matters, and it should be pretty trivial. So that's our schedule. Then we have so we have a nice announcement here, the open source blog at Google. They mentioned that, there were the acceptances. Let's celebrate. There were 43,765 applicants from 160 countries, 7,723 proposals submitted, 967 contributors accepted from 65 countries. So that's nice to see a lot of worldwide diversity there and a lot of people being part of this program. And then I want to point out the mentors. So Jiahang is a mentor for this project. He's here. Mayuk Deb, who's not here, is also a mentor. And so you'll be going to either Mayuk or Jiahang for questions. Jiahang, of course, is his focus is the graph neural networks part, and then Mayuk is his part is the DevoLearn part. He's sort of the maintainer for DevoLearn. And the reason we're doing it that way is because we want to bring a lot of this stuff together this summer. We want to do the graph neural networks part. We want to improve the segmentation part. And then we want to have like some sort of a unified package where people can come to DevoLearn. They can do the segmentation in different ways. They can do the graph neural networks. And, we want to make that usable for people, so it's not like they have to dig through a lot of documentation to use it. And so I hope by the end of the summer we can get to a point where it's really, looks like it's really, coming along. So that's our goal for the summer. And so that's great. And we have over 2,400 mentors. Jesse, by the way, is also a GSoC mentor in a different set of projects, but congratulations to all the mentors. I've prepared a blog post on this. So this is the Google Summer of Code 2023. This is a blog I've been running for over 10 years. It's got a lot of content on it, but this is just the post describing our. So these are the projects I'm mentoring. I'm mentoring 2 students through DevoWorm, and then I'm also mentoring people through the Orthogonal Research and Education Lab. And Jesse, of course, is involved in that. And Himanshu actually was a GSoC student in the Orthogonal Lab last year. So he's actually been through GSoC already. So congratulations to everyone. So this is our community. Our community consists of OpenWorm, DevoWorm, and then to a lesser extent, the Orthogonal Research Lab. So OpenWorm is this is the website for OpenWorm. There are these different projects like Cybernetic, which is like a biophysics simulator. So they can simulate the C. elegans in different environments and on different substrates. And so they're looking at how the C. elegans moves against different substrates. They're looking at the biophysics of the organism, the muscles, the soft tissues, and the fluids that they encounter. So there are also other things like WormSim and, of course, DevoWorm. The OpenWorm browser, which is like a 3-dimensional browser that you can open up or a 3-dimensional viewer that you can open up in the browser. And there's an initiative to bring this to virtual reality. So, this is a model that kind of puts together the morphology of the worm, models every neuron, and models the connections. So you can explore that. You can also simulate those connections with something like NeuroML, the Connectome model, or, Geppetto is a thing that models individual cells, especially their electrophysiology. And then there's other things like C302, where they put that together in a connectome. So there are a bunch of different projects that have come out of OpenWorm. And so you might explore a lot of what OpenWorm has. There's a lot of maturity in some of these projects. There are also things like the Movement project, which I mentioned before, where they took pictures of images of C. elegans on different surfaces, different genetic mutants of C. elegans, and they characterize the movement dynamics. So a lot of those materials are in this repository. And this repository is from an open house in 2016 that we had, where we had everything, all the different subgroups come together and give presentations. So some of this material is dated, but it should give you a good idea of what's going on. So there are all sorts of different things going on in OpenWorm, from different types of tools to different types of modeling paradigms to different types of approaches. So I'll put this link in the Slack, and you can have that as sort of a way to explore OpenWorm. There's of course the rest of the Slack, which if you're in the Slack, you're in the DevoWorm channel. You go outside the DevoWorm channel, and there are a lot of different channels to explore. Some of these are project-based. Some of these are, maybe organizational based. There isn't a lot of activity outside of the DevoWorm channel and the Slack, but there's a lot of stuff that's archived. So you'll be able to learn something about that. DevoWorm itself. So DevoWorm has a website as well. This is our website. We have different links here. We have people. We have our educational initiatives, our media and public lectures, which are different talks that have been given. There's, we have DevoWorm ML, which is a machine learning course that we did several years ago, sort of bringing together a lot of these deep learning, machine learning techniques in biology. And then we have Google Summer of Code, which gives you information about our prior Google Summer of Code students. We have publications. We have different data sources. We even have demos, which needs to be updated, but that's another topic. Our affiliations, and then our schedule. So, there are a lot of resources here at this site, devoworm.weebly.com. I'll put that in the Slack as well. We have a YouTube channel, which has actually become quite important. So the YouTube channel has a lot of the meetings and talks that we've had in the group. We've had tutorials, we've had academic lectures, we've had recap talks. This one here, Digital Diatoms, is one where I went over the digital diatoms research. So we work not just on C. elegans, but we work on diatoms and other types of simple kind of organisms. I don't want to say simple, but basically single cell or a small number of cells. We're also interested in things like axolotl and other types of embryos that are more complex, I guess you could say. And then we're interested in computational models and digital models. So, we're interested in physics, cell physics, and things like that. We'll have to make more of these recap videos, but I think there's a playlist where we kind of go over some of these things. So there are a number of playlists. You can also find videos that way, and that really does recap a lot of what we've done over the years. So that's the website, that's the YouTube channel. We're also involved with the Orthogonal Research and Education Lab, which is a separate organization. We're doing things like working on sort of computational neuroscience models, working on different things with open science and open data, working on different informatics topics. And so this is something that we may, reach out to from time to time for resources. But just to make you aware that that's there. So that's our community overview. So the community just means people that you might be able to draw from in terms of resources. So if I go to, if Himanshu wants to find some resources on C. elegans movement, there are resources in our community we can incorporate. There are data sets also, but I'm not sure. There was a movement website, but I think it's no longer operative. So they're different, there are different people you can connect with. They can provide different resources. And that's one of the points of the community period. The other point of the community period, and this will be something I'll talk about next time or the time after that, is that there's this whole issue of creating open source software. So at the end of your projects, you'll be creating software that's open source. You'll have a license, you'll have some things. And then the point here is I want to be able to help you understand how to go through this process of creating open source software, licensing it, and then understanding how to do that, so that if you go on in your career and you build open source software, you can do that effectively. So we have something Dick put in the chat here. Picket heaps, life in glass houses. So what is this?
This is the best movie to introduce people to diatoms.
Oh, okay, yeah.
Okay, and we've created the subtitles in English, which is a lot of work.
Yeah.
Okay, and we'd like to get volunteers to translate it to other languages so it's widely distributed. Picket heaps died about a year or two ago. Okay, so this is sort of his legacy, but it's also a fantastic movie.
Yeah.
Okay.
Yeah.
And it's being published online as a supplement to a journal. It's freshwater and knowledge or something like that. Okay.
Yeah. Okay, that sounds good.
So if anyone wants to help translate it to another language other than English, we'd be glad to have you do it.
Yeah. All right. Thanks for that. So Susan, or you said you may have some pictures to show, or? Oh, you're muted, Susan.
I have to do things in order. There we go. Share screen. Can you see my screen?
Not yet.
Okay. This again. Okay. Oh, there. Select the tab to share. Okay.
Or sometimes the whole screen works too.
Entire screen. I'll try that. Okay.
There
There.
we go. Yeah, there we go.
All right. Let's see if this works. So, can you see my eggs?
Yeah, I can see them.
Thousands of pictures. There
Yeah.
we go. Now, can you see that the single egg here?
Yeah, I can make it out. Sort of the edges of it.
Okay, because you can actually see the individual cells here.
Oh yeah.
And
So
there, can you see that?
Yeah.
Okay, well these are from my microscope, and I took several pictures from each view, sometimes with more lighting, sometimes with less, in and out of focus on various parts. So hopefully we can get a good view. Now this is the top at an angle, and I turned on the light on the microscope, and you can see the top cells easier with the light on, but it obscures some of the cells. So then I took images with less light, so you can see where the cells are, where the light was, if you know what I mean.
Yeah.
And so there's another view. Some of them are obscured by the surrounding gel, and it might be that I should take the gel completely off to image these, but I would need a little cup holder. So these are just stuck on the slide. I just put them on the slide and took pictures of them.
Wow.
So that's what these are.
So what is the which microscope is this? This is the ball
This
microscope.
is my ball microscope.
A ball microscope. Okay.
Yeah, I think I need a different camera for the flipping microscope, and that costs a couple thousand or 3,000 Canadian. So it's not happening anytime soon. So anyway, so this is there. Now this division of cells here looks interesting. Can you see the middle division of cells?
Almost looks hexagonal, actually.
Yeah, and this is where the later in development, the cells start. It starts contracting into the bottom. What do you call that? I should know. Anyway, it looks like it's in the same position as these cells are dividing. So it's sort of interesting. Anyway, this is a better picture, but I need to make it larger.
Yeah.
I just took pictures. I tried to fiddle with the size of things, and I got into an argument with my microscope and decided pictures are better than an argument. So I just took pictures.
So all this around the edge is gel you have to put into the...
Yeah, there's gel around the edge of most of these.
And some of these are really good, yeah. I mean,
there.
When you go to time lapse, what's the range of intervals for the time lapse you can get?
I can make it fairly fast. Slower is more of a problem with the Arduinos. And I was running into trouble getting them to stay in focus, some of them. I guess it's just I'd have to make sure the best microscopes were in the slots. and then it would work. And yeah, and it changes because the gel dries out. So I think putting them in a little cup holder would be best so that the focal range doesn't change. Yeah, just tell me what you want in the way of timing, and I'll try to do it.
Okay, those reflections of the LEDs are off of the gel.
Yes.
Okay, so taking it out of the gel would possibly remove them.
Probably they would reflect off of especially the yolk on the bottom.
Okay.
Yeah. So I've got a whole bunch here. This is the first set, and I was having trouble focusing on it because it doesn't have any divisions yet, and this is sort of early. And the other problem I had was the microscopes themselves make these shadows on the surface. So that's actually a shadow of a microscope. So I need to get external lighting on them from various angles so there's not so much of a shadow. Anyway. But yeah, I have lots and lots of images.
Yeah.
You can see that each one of these has like multiple.
There's a general problem here, Bradley.
Yeah.
Any segmentation algorithm has to also figure out which is artifacts and which are real cells.
Oh, right. Yeah.
Okay.
This is a prime example. This is a microscope shadow with light, and this is a microscope shadow. That's a microscope shadow, but it's dividing in two. I finally got a nice picture of this egg dividing in two. This first cell division. Oh,
yeah.
that was nice. There it is.
Okay. Yeah.
You still see the shadows. So
yeah.
You can see the shadows. So you'd have to get rid of the shadows. But I mean, they're fixed. They're there throughout the whole series of pictures. So. Yeah, so I was going to try to fix the lighting so that wasn't there. This is starting to divide into the second division. So there's first division, second division, so it's dividing in 4. Here, these are early pictures. I did better with the later ones. So you want to see later one? I don't know. Started. There's the bottom of it. Oops. More cell divisions. Yeah, this is, yeah. Oh, there. Sort of. But you can still see the shadows. But that one doesn't have shadows on it. So yeah, lots and lots of pictures. I could show you pictures for an hour or so, probably, because all of these folders have at least, well, a lot of them have 30 images in them, and that, yeah. Just, and I so far done four eggs, imaged them. So
that's great. Yeah.
So do you want to see any more, or is this good, or?
Yeah. So you have them for like how many stages of development? Like you have basically for.
Well, I just sort of took them whenever the spirit moved me,
Okay.
because I'm working on my other project. So see this, I have 15 or 16 images of this. So it went from just a few cells to this. And this is what I was talking about, that cell division that was in the bottom. This is at the bottom too.
That may be a contraction rate rather than a cell division.
No, this is a contraction ring. The other previous one was a cell division, and this is a contraction ring. They're kind of in the same spot. I don't know, you can't see individual cells very well here. You can sort of see them.
Are these wild type or
wild type?
They're wild type, okay.
Yeah, they're the ones that survive the best, so that's kind of what I have. But I do have one leucistic or white salamander. Anyway, this is the ball microscope.
Yeah. So for Himanshu and Jiahang and Jesse's benefit, the ball microscope is where you have the embryo on like a stage or a central location, and then you have microscopes from different angles. So 9 or 10 different angles. And you're getting information from all of those angles.
Okay, well, I'm just going to minimize this and see if I can help you with some of that. Show off my dirty living room here.
Oh.
So there you go. There's the ball microscope.
There it is.
And I've been plugging it into this computer. And each one of those USB cords, I plug them in one at a time to get the images. And there's my coffee cup.
So you do like 9 or 10 inputs into a single.
There's 10. There's 10.
Okay, 10. So
in a row.
And so you have 10 channels of. Yeah.
I don't know if you can see that, but there's an egg.
Yeah. Oh, it's kind of out of focus, but yeah, it'll be. Yeah, it's basically you can see the egg a little bit.
It's tiny and it fits into.
It's not so tiny. It's 2 millimeters.
Into that right there.
Yeah.
Right on top there.
Yeah.
And then.
Oh, I see. It's because the microscopes are so close that you can get shadowing.
Yeah, you get shadowing, and then you put the top on and line it up with the screw here. Screw.
So either you have to correct for the shadows, or you have to get enough light in so the shadows are very low.
That's right.
Yeah.
So there we are. See?
Yep.
Anyways. Okay, then this is not good. There we go.
Yeah.
Sure. Anyway, that's what I've been messing with this week, and that's why I haven't got my 12 graphs done for my presentation at 2. So we need to keep on working here.
Yeah.
Just don't tell them.
Yeah. Priorities,
Yeah.
Yeah, that's great though. I mean, I'm glad to see that. Yeah, we're getting. I always like to see data like that.
Yeah, so I can send this to you.
Yeah.
They're small, like they're 640 pixels times 420 or something. So they're not as high a resolution image as one might like, but I would like 14 megapixel cameras instead of 2 or 3 or whatever it is. But they didn't have them. But if they did, this would be really rocking. It was correct.
Yeah.
So yeah, that's what I have so far, and I can keep taking some images. It's hard to get the salamanders to lay in. I only have 2 females that can lay eggs, and I'm not too sure about the second one.
Hey, Susan, is there any prospect for motorizing the focus on each microscope?
That would be more expensive.
Oh.
You probably figure something out. I'm sure I could find a nice $2,000 piezo movement.
Oh, okay. Yeah, because this is a good case for automatic focusing.
Yes. Yeah. Yeah, it would be nice. But I think I can put them in focus, and if it's a good microscope, not a loose one, I have a couple of loose ones.
What about the prospect of putting the embryo on a 3D stage? It can move.
Oh yeah, the news is that Kent has sold a microscope to the University of Manitoba Geology Department to look at dinosaur tooths. And they might let me in there with a little 3D stage, and I could take images of salamanders.
That might be easier because you could then fire the microscopes in order, but focus each one.
Oh, well, no, this is the Kent's microscope is like this, and then it tilts. So I would be able to get a picture of the top or the bottom of the egg.
Yeah, what I'm talking about is putting a tiny stage which has piezoelectric motion in 3 axes. And use that to hold the embryo.
Okay.
Then you could focus the embryo at a given camera, take a picture, and then go to another microscope and refocus it there, moving the stage instead of moving the microscopes.
I don't like to move the embryo.
Well, you may not like to, but you may have to.
But when it moves, and then it jiggles for a while.
Okay, what you do is you put it in a methyl cellulose solution, which is probably harmless.
Well, or leave it in its gel and get it to attach properly. But it still creates that capsule around it. So
I know. Well, yeah, but if you take the capsule off and put it in methyl cellulose itself is viscous and harmless.
Yeah, I'm aware of it. Okay. It's just that it still will create a pocket of water around itself. Water.
Okay, but there'll be a settling time. Okay, but you can account for the settling.
Yeah, that's okay. Yeah, the flipping microscope, after all, does work. In spite of being flipped.
Right.
So, yeah, I don't know, for the Kent's microscope, the flipping microscope might work too. But you could put the flipping stage in the Kent's microscope.
Okay.
Yeah.
Oh, that's automatic flipping?
Yeah, I've had it automatically flipping.
Oh, okay, yeah, because when we did it at first, it was hand flipping.
No, this is automated flipping. Yeah.
Okay.
It's I'm not sure about the angle on it because the rubber, my rubber wheels are not gripping like I wanted, but for sure, just flipping works. And maybe a 3D stage or circular stage that turns, circular stage might work better.
Okay.
So just leave the flipping part and then turn the stage and take pictures with the Kent's microscope from angles.
Okay.
That'll get the top part or the bottom part, depending.
What do the geologists use it for?
Well, I could say dinosaur teeth.
Oh, I see. So they're not using it for crystals or something. Okay.
Oh, maybe. I don't know. It's an expensive acquisition, and they haven't got it yet and stuff. And the guy from Kent said he wanted salamander eggs to take pictures of with the Kent's microscope, and so I sent him some. I don't know if they'll arrive alive or not, but I sent him some.
Okay.
Yep. These are axolotls from Kijiji. They're mine, and so anyway.
Okay.
All right.
Yeah.
Well, that's great. Yeah. Yeah. Thanks. Yeah. Thanks for showing the microscope. So Jesse said so. Yeah. So Yeah, I think that's it for today. If you had anything else you wanted to mention before next week, is I said, Himanshu and Sushmanth, we should keep, in communication on Slack. So, I don't expect a daily update, but if you have anything you want to share, we have our channels, we have our DMs. Just make use of those. And then, weekly we'll be having a meeting where you should give a short update on what you've done the last week. And then towards the end of the community period, we'll have a presentation where you can talk about your proposal, what your plan is for the summer, and then at the end of the summer we'll have another presentation to show how that panned out. So then that's pretty much what we'll do this summer in terms of meetings and communication. Yeah, much. Thank you. All right.
Okay. Bradley,
Yeah.
I sent you another version of our paper this morning.
Okay.
You got a second to discuss it?
Yeah.
Okay.
All right.
Okay. It can be after the meeting.
Yeah, after. Okay. Thanks, everyone. Take care.
Bye.
Bye. And now I'd like to talk about a new thing we're exploring within the group. It's something called assembly theory. So we're working on a paper and some research on the area of early life. And one of the issues in early life is that you have Darwinian evolution that you can trace back to a certain point in time. But before that point, things have to assemble from non-life. And so the question is how does that happen? It requires a new theory because we can't necessarily use Darwinian evolution to describe the transition from non-life to life. On the other hand, things don't just kind of show up. They have to assemble from basically biochemistry. And so how does that process happen? So this is a paper that it's actually an article that points to some papers that I'm going to cover. It's by Philip Ball, who's a science writer. And the title is A New Idea for How to Assemble Life. So this is a newish theory that has been developed called assembly theory. If we want to understand complex constructions, such as ourselves, assembly theory says we must account for the entire history of how such entities came to be. And so you can see there's this network of Lego bricks in this image that sort of explains what they're trying to get at. They're trying to get at this idea that things are assembled, at least a minimal living system. And then the minimal living system can evolve from there. So they have this assembly of this little Lego person. Of course, that's not exactly at the scale of complexity that we're getting with assembly theory, but that's for the benefit of the image. Assembly theory explains why, given seemingly infinite combinatorial possibilities, we only observe a certain subset of objects in our universe. So basically the idea is that there's this immense combinatorial space, that there are different ways that these things can be combined, but in terms of plausibility, only a certain subset of objects are possible. And indeed, those are the ones that emerge to become life. So not every combination is equally plausible. Life on other worlds, if it exists, might be so alien as to be unrecognizable. There's no guarantee that alien biology would use the same chemistries as on Earth with familiar building blocks such as DNA and proteins. Scientists may even spot the signatures of such life forms without knowing they're the work of biology. This problem is far from hypothetical. In April, the European Space Agency's JUICE spacecraft blasted off from French Guiana on a course to Jupiter and moons. One of these moons, Europa, has a deep briny ocean beneath the frozen crust and is among the most promising places in the solar system to look for alien life. And so basically, the search for life suffers from a fatal flaw, which is there's no definite signature of life. So you can search for the signatures of life we know of on Earth. We can go to deep sea vents and look at some of the things that are going on there. So we can look at a number of different things on this planet in terms of the life that exists on this planet. But if we go to another planet those processes might be driven by different chemistries. And so we don't necessarily know what is living and what is not. So one of the things that this theory tries to explain is what those signatures should look like, what plausible forms of life should look like, and not necessarily be biased towards any one particular chemistry. So this was led by Lee Cronin and Sarah Walker. They propose a very general way to identify molecules made by living systems, even those using unfamiliar chemistries. Their method simply assumes that alien life forms will produce molecules with a chemical complexity similar to that of life on Earth. And so in last week's meeting, we talked about the Miller-Urey experiment, where they applied an electrical field to a bag of chemicals, a chemistry that had some potential, and they were able to generate a lot of different macromolecules that were lifelike. And so this experiment has been done again and again. So we know that early life had a lot of electrical potential, a lot of heat energy, a lot of lightning. And so we know that was abundant in that environment. And these chemistries are also abundant in different places. And so put that together and you get life, and probably starting multiple times. And it's not clear necessarily how that coalesced in the tree of life. But this is the problem we're going to face on another planet, say, that they would have a chemical complexity. There would be some event that gives birth to life, probably multiple such events, and then that emerges into some sort of tree of life for that planet. Called assembly theory, the idea underpinning the pair's strategies, even grander aims. So they have a series of publications. They have 3 publications they link to here. The first one being this one, on exploring and mapping chemical space with molecular assembly trees. This is in Science Advances. This gives an idea of this sort of combinatorial space and what that looks like. There's another paper here that is exploring the sequence space of unknown oligomers and polymers. So this is, again, exploring that combinatorial space. And then the third one is formalizing the pathways to life using assembly spaces. So this is considering that assembly space and then considering that pathway to life being this transition from non-life to life. And so this theory attempts to explain why apparently unlikely things, such as you and me, even exist at all. And it seeks that explanation not in the usual manner of physics, in timeless physical laws, but in a process of it imbues objects with histories and memories about what came before them. It even seeks to answer a question that perplexed scientists and philosophers for millennia. What is life anyway? So this is something that, it's not without controversy. So there's no clear path to testing this in the lab, but there is this clear path to a set of computational experiments and some sort of characterization of this process of assembly. So the thing we're interested in is not necessarily like any specific path to life, but of the plausible paths to life. And so those should emerge out of this search of the combinatorial chemical space. So an assembly theory involved in part to capture Lee Cronin's suspicion that complex molecules can't just emerge into existence because the combinatorial space is too vast. So the principle here is that these plausible paths through the space are driven by constraints, but they're also driven by opportunities. And so we don't really know how these pathways emerge. We just know that some are going to be more plausible than others, and that's based on the factors unique to that setting. So on Earth, there may have been a certain set of conditions that led to the emergence of life. On another planet, you might have another set of conditions. They don't have to be identical to Earth. They can be different. Planetary history, the chemistry of the life form, those are all different, and so that's going to not only yield a different combinatorial space, but a different set of plausible pathways. So life on Earth, for example, may not be very plausible in a place like Europa, but it still may have life. So this may bring a fresh perspective to this problem of early life. We have our own ideas about early life, and so we've stated these in this paper we're working on. So when this paper is done, we'll present it in the group. But this is a way to sort of think about complexity and get your hands around this problem. And so a fun little figure that I found, and this is actually related to this particular article, is an experiment that they did where they tried to estimate the assembly index of various substances by repeatedly measuring their molecular structures. So out of assembly theory in this set of pathways arises this assembly index. So how do you assess these pathways and their success or their failure? And so one way to do this is to use this assembly index. So this is for various substances by repeatedly measuring their molecular structures. So you look at the molecular structure, you look at how complex it is, you see how many parts there are, and that's an assembly index. And we've done actually something similar in this paper using Braitenberg Vehicles, which is a model typically used in robotics or in cognitive science to explain like embodied intelligence. In this case, we're using it as the assembly of early life forms that are viable, that are functional. And so this is a similar method where you're using chemical structures and you're looking at the complexity of those structures in different ways. And so they come up with this assembly index, and they compare this over a number of substances and actually biological systems as well. So we have in a non-biological realm, we have coal, quartz, and granite. And so these are, coal comes, of course, from organic materials. Quartz and granite do not, but they should have, you would think, what is the complexity? How do you, you have a common framework to compare them. How do they compare? So coal actually has, so low here is 10 to 12. So there's like a range of values here. It's like, this is the low value, this is the high value, this is the average. They just give the numbers the low and the high. So coal is a low of 10, a high of 12. Quartz, and so remember coal is derived from organic materials. Quartz, on the other hand, has a low of 11 and a high of 12, which means that you have this, that the quartz may be a little bit more complex. Granite, however, has a low of 10 and a high of 15 with an average of 12. So that means that granite is maybe even more complex than coal or quartz, despite being a rock versus like something that derived from organic materials or another type of rock. So, clearly there's some, variation here. But these are all non-biological by and large. So they don't really, they're not living systems. Now you can look at living systems as well. And so here we have yeast, urine, seawater, E. coli, and beer. And of course, beer is derived from yeast. So there's this aspect, again, like coal, of being derived from a living system or biological system, organic system. So we can see here that all these biological systems are clearly higher in complexity than the non-biological ones. So if you compare it with rock, with stone, something like yeast has a low of 10 and a high of 24 with an average of 14. So you see that the low and the average assembly index aren't really that much different from non-biological systems. But you do get these high levels of complexity that, and I don't know how they measure this, if they're measuring behavioral states or different conformational states of proteins or what. But we can see that there's a high-end measurement here. The same thing with urine, which is urea and water and things like that, where you go from 9 to 27 with an average of 14. So again, you have a higher complexity. And of course, urea serves a number of functions biologically. It also carries waste when it's suspended in water and so forth. Seawater, which of course has a lot of microorganisms in it. It's, not organic in and of itself, but it has harbors a lot of life. and DNA and so forth. Seawater has a low of 9, a high of 28, and an average of 13. So it's somewhat like urine, somewhat like yeast. E. coli is, of course, organism. You're sampling a single cell organism with DNA, proteins, and the like. It's probably more concentrated than what you'd find in urine or seawater. Urine also carrying maybe some microbial life. But E. coli has a low of 15, a high of 31, and an average of 20. So clearly this is, we have a living organism versus rock. The living organism has a much higher assembly index. And then beer, which is derived from yeast, has a low of 10, a high of 34, and an average of 16. So these high measurements are really interesting because for living systems, they're very, very high compared to non-living systems. And so, the question arises, what is contributing to this, especially when we see that the low values are all sort of similar. What does it arise from? And so this is really, I think, showing that the assembly index is measuring something about biological complexity and livingness. We don't know exactly what, at least I can't tell from this figure, but that's work for future papers and theory and observations. Okay, now I'm going to talk about one of the papers in particular that we're linked to from that Quanta article, and I'm going to compare it to some of the work we've been doing with the Braitenberg vehicle toy models. So this paper is exploring and mapping chemical space with molecular assembly trees. And so they use a tree of steps to show the path through this possibility space. So we have an example here in figure 1 where you have representations of an assembly pathway. So you see that step 1 is where you have these molecules that are dissociated. Step 2 is where they come together in a bond. Step 3 is where they have another bond come together, and then step 4 is where it forms an adenine. So you get these different steps where things are assembled. And then this leads to some sort of thing that's biochemistry from a simple chemistry. So you have this, toolbox of things that get put together in stages, and you end up in this situation where you have a biomolecule after a number of assembly steps. So there are these pathways that come through, that are more plausible than others, given the nature of chemical bonds, the different electrical charges, and so forth. So, so the colors show these building blocks. Here you have this yellow and this pink that show the building blocks. You have a toolbox of building blocks. You put them together, and things get assembled a certain pathway. So that's showing basically what they're talking about when they say assembly theory. You're assembling things from smaller parts. So this is a molecular assembly tree where you have this path through a possibility space. There are a number of possibilities for assembling things, and you get the path. This still doesn't really show the tree explicitly. But if we go down to this figure 3, it's a little bit clearer. You have these building blocks. You have these paths to different things, ADP, ATP, and AD+, different bases of a RNA molecule, actually a DNA molecule, then you have RNA and thymine from uracil. So, all right, so this is the 4 bases of an RNA molecule. You have a uracil gets converted to thymine, and these are the 4 bases of the DNA molecule. So you can see how that works. And again, these are all necessary for a living system. They all emerge from non-living components or building blocks, and those in turn emerge from inert molecules. So this is not, there's no line that says this is living or non-living. It's just this pathway to success. And so if we turn to our toy model, I've worked out an example using Braitenberg Vehicles, how this might work, with our toy model. So we have these assembly pathways. We have different rules of assembly. This is we had in the assembly tree, and we can see how this works. So you have a vehicle body, a single panel of a vehicle body. You start with that. You start with a number of those. You add another part. So there's a rule that says you can duplicate the single panel. So you have two panels. Those get put together as you see at the end. And then you have these wheels, which are the four things on the outside. Those get put together, and those two steps, duplication and addition, you're duplicating this part, you're adding these parts, and you get this. This is a viable vehicle because it's bilaterally symmetrical. It doesn't have too much space where there's no weight-bearing aspect to it, and it can propel itself. So in this case, what I'm showing you here is just a viable outcome for these assembly rules and these assembly steps. But let's suppose that that's not the case. Let's suppose that we have an asymmetry. So we have a panel, and we have a rule of duplication. So you have two panels that kind of align, top to bottom, or I guess anterior, posterior. And then you have not four wheels, but three wheels. So you add three wheels with addition, and you end up with this asymmetrical thing that isn't functional, or not functional enough according to our criterion. So this is this circle with a slash through it. But the thing is that if it's close enough to being viable, you might actually have the opportunity to rescue something. So this is all non-living stuff by itself. These things are added together using their rules, and we end up with this non-viable vehicle. But it's non-viable. It's not completely non-viable. It's somewhat non-viable. And so it's possible to rescue that function using a flagellar rotor, which we've added here in step 3. This is another addition rule, and now you have a viable vehicle. And so according to our criterion, it's now functional. And it's not chemistry, but it's this vehicle toy model example where you get this transition from non-living to living, where in this case, it's non-functional to functional. And so this is just an example, moving from sort of the hardcore biochemistry to something that maybe straddles living, simple living organisms / behaving organisms. So just wanted to show you that.